Good evening everybody, and welcome to BrisScience for April 2023. Thank you so much for coming out tonight. My name is Joel Gilmore and I am your MC for this evening. And thank you so much for coming along. BrisScience, if you don't know it, is a series of free public lectures on science held once a month by the University of Queensland here at the fantastic State Library of Queensland, our wonderful venue partners for so long now. And we aim to bring not just the best scientists, but also the best communicators to share their research and the latest cutting-edge science with you. So I would like to start by acknowledging the traditional owners of the lands on which we're meeting tonight and pay my respects to elders past, present and emerging, and to acknowledge that ongoing contribution that the first people of Australia make to our understanding of how to live on and care for this land, and to learn all about it through our other scientific endeavours. So, great note to start this evening on. Okay, if you have a phone, now is a great time to put it onto silent, but do not put it away, because after our presentation we will be taking questions over our question app. Look at that perfectly timed QR code right there. You can scan that QR code or enter the bit.ly link there directly. Put your questions in over the talk and at the end, and then we'll get through as many of those as we can. But there will also be lots of opportunity for you to engage both with yourselves and with our speaker afterwards, because we'll be having food and drink outside. So we really encourage you to stay around and make this a night of science and culture. Okay, I think that's all the housekeeping we need. So to get started, it's a little known fact, perhaps, except for maybe some of our regulars, that a few years ago at BrisScience, we held a talk called Pathogens, Pandemics and Preparing for Disease X. 3 months later, the COVID pandemic started. Therefore, it is with some trepidation that I tonight welcome you to a talk about viruses and vaccines. But we've all got to take some risks in our life, so I'm going to roll with it. Tonight we are talking particularly about viruses from mosquitoes and how perhaps some of the benevolent viruses might be co-opted into helping us tackle those viruses by developing new vaccines or even new drugs. To talk us through this tonight, who better than Dr Jody Hobson-Peters, a virologist from the University of Queensland who specialises in, you'll never guess, mosquito diseases, amongst other things. And so to help us swat up on our mosquitoes, thank you, and cure our thirst for knowledge, please put your hands together and welcome Dr Jody Hobson-Peters.
Hello, so thank you so much for the opportunity to speak about my research tonight. Now, as for any research program, the story that I'm talking about tonight is the culmination of many years of work, and also the efforts of a number of researchers, not only at the University of Queensland, but also at other institutes around Australia as well. And so I must, however, acknowledge Professor Roy Hall, who has been my mentor and my boss for many years now and has recently retired from full-time academia, but he was instrumental in starting the program that I'm talking about this evening. So, as it was just said, I work with mosquito-borne viruses, and the viruses that I will be talking about tonight are those that are transmitted by arthropods. So these viruses may be transmitted by ticks or sandflies or mosquitoes, for example. And you'll see on the left-hand side there are many significant pathogens that are significant for both human health and in some cases animal health as well. You may be familiar with some of these viruses, for example, Zika, dengue, Japanese encephalitis, also Ross River virus is up there as well. So these viruses must be capable of replicating in the arthropod vector. Most commonly for a lot of these it is a mosquito, but also in an amplifying vertebrate host. So they must be capable of replicating in an animal. And so the transmission cycle is when a mosquito that is infected with these viruses bites an animal, and then in this animal, depending on the virus that we're talking about, whether it be a bird or a pig or a human, for instance, these animals then replicate the virus to sufficient levels in their bloodstream to subsequently infect another mosquito that then bites them. And this is how the transmission cycle occurs. However, the viruses that I'm going to be speaking about tonight are the insect-specific viruses. And these viruses probably these days should be more accurately classified as mosquito-specific viruses, because all of the viruses on the right-hand side of the screen are those viruses that are restricted to replicating only in mosquitoes. So these viruses are maintained exclusively within the mosquito population without any ability to replicate in vertebrate cells or in animal cells. Now, what you may also notice is that many of these viruses that are insect-specific or mosquito-specific are within the same viral families as many of the pathogens on the left-hand side of the screen. And this means that they are indeed genetically related to these pathogens. And so the hypothesis is that these insect-specific viruses are likely the ancestors of those viruses that then became pathogenic to humans and other animals. Now tonight I'm going to be speaking about a very special virus that we discovered back in 2010 called Binjari virus. It is a flavivirus related to other pathogenic viruses such as Zika and dengue. And then I'll also be giving a very quick, sneak peek at some more recent work that we've been performing on using an insect-specific alpha virus that's also from Australia called yada yada virus. So just to reiterate the difference in these transmission cycles. So if we're looking at the flaviviruses, of which dengue, Zika, West Nile Japanese encephalitis are all flaviviruses, they must be capable of replicating in the mosquito and being transmitted by the mosquito to an amplifying vertebrate host. However, the insect-specific flaviviruses, on the other hand, as I mentioned, maintain exclusively within that mosquito population and don't replicate in vertebrate cells. And so for them to be maintained in the mosquito population, there is an incredibly strong mechanism of vertical transmission, meaning that these viruses are transmitted from the mother mosquito to her progeny, most likely via an infected egg. So back in 2010, we started this virus discovery program, and I must admit that a lot of our colleagues were very perplexed as to why we would be going down this research road. Because these viruses, at that point in time, not much was known about them, and most of the research was dedicated towards understanding viruses that did actually cause disease in humans and animals. But the reason why we chose to start studying these viruses was, firstly, we believed that the study of insect-specific viruses would provide us with clues as to the evolution of pathogens. We were also very interested in understanding how the presence of these viruses within mosquito populations was affecting the transmission dynamics of the pathogenic viruses. And importantly, we were asking the question, can we harness the really unique characteristics of these insect-specific viruses to our advantage? Could we potentially use them as natural biocontrol agents, or can we use them in biotechnology applications? And this is the story that I will be telling tonight. So to start off our virus discovery program, we already had a lot of mosquitoes stored in our freezer, a lot of archival mosquito pools, but we also had an extensive network of collaborators over Australia as well that were actively collecting mosquitoes in mosquito traps like that on the left-hand side of the screen. Once these mosquitoes are trapped, they are then sorted into the different species of mosquitoes, after which we essentially grind them up. And after we grind them up, and I'm sure everyone likes to do that, right? So yeah, we get a lot of satisfaction out of that. Grind them up, and then we inoculate them onto mosquito cells in culture in the laboratory, and then we see if we can culture any viruses out of these mosquitoes. And so that's essentially what the images at the bottom of this screen are. Now, a lot of these viruses, we didn't actually really know what we were looking for. This research area was still very new, and so we didn't have any tools to detect these viruses, and so we had to develop some really broad spectrum diagnostic assays to be able to detect the replication of these viruses within the cell cultures. And so if we got positive hits for some of these virus pools, then we went on to sequence the genomes of the viruses to try and identify what families that these viruses belong to. So as you can imagine, we discovered a huge array of different insect-specific viruses in the mosquito populations collected from all around Australia. And I guess importantly, what we wanted to prove initially is that these were indeed insect-specific. And to do this, we inoculate them onto cultures of cells. And so we've got lots of different cell lines in the laboratory whereby we can take the viruses and infect them onto these cell monolayers. And these cells may be derived from mammals or from birds, reptiles, amphibians, for instance, and we proved that none of these viruses were capable of replicating in any of these cell lines, so all of these insect-specific viruses here. However, our viruses that typically are pathogenic, they do replicate just nicely in all of these vertebrate cell lines that we assessed. But of course, these being mosquito-derived, all of these viruses did grow really well in insect cells, in mosquito cells in culture. And what I also want to highlight here is that all of these viral species were brand new, and a lot of them were unique to Australia. So today I will be focusing on the flaviviruses, the insect-specific flaviviruses that we discovered. So the insect-specific flaviviruses are grouped into two main lineages. So we've got our lineage 1 insect-specific flaviviruses here that are a lot more genetically different to our vertebrate infecting flaviviruses down here. So we've got Zika and dengue, West Nile down here. So we actually found quite a number of these insect-specific flaviviruses belonging to lineage 1. But it was actually those viruses from lineage 2 here that are a lot more genetically closely related to our pathogens here that we were really interested in. And at that point in time, not many of these viruses had been discovered. We had, however, discovered a very tiny amount of sequence of a virus that we subsequently named Binjari virus in a mosquito pool that was collected in the Northern Territory. Now, unfortunately, that particular mosquito pool hadn't been kept cold enough, and so the virus died in that mosquito pool. So we were never able to recover an isolate. So an isolate being something that we can culture up in the laboratory. And so we tasked our PhD student at the time, Dr. Jessica Harrison, to screen thousands and thousands and thousands of mosquitoes, trying to get a viable isolate of Binjari virus. And she did end up succeeding, but it did take a lot of years. So in the end, it wasn't until the end of 2016 that we finally got an isolate of Binjari virus. And this was also collected from a mosquito pool in the Northern Territory. And as fate should have it, as soon as we finally got this elusive Binjari virus, that we isolated a very close cousin of Binjari virus, which we subsequently named Hidden Valley virus. And the interesting thing is that this virus, the mosquito pool that this virus was taken from, was taken in 1976 and had been stored in a freezer in Western Australia since that time. So now that we finally had a viable culture of Binjari virus, we began to look at the characteristics of it, and we found that it had a number of very desirable characteristics. The first of these being that it does not replicate in vertebrate cells in culture, so it was definitely restricted to replication in mosquito cells. Secondly, we saw incredibly high replication of this virus in mosquito cells in culture. So here's Binjari virus and its close cousin, Hidden Valley virus. And so we're seeing up to 10 to the 10 infectious units per mil. So this is 10 billion virus particles in every milliliter of culture supernatant in those mosquito cells that we culture up in the laboratory. And importantly was this virus's incredible tolerance for the exchange of the genes that code for the structural proteins on the outside of the virus particle. So these are the PRM and envelope genes that form the outer case, if you like, of the virus particle. So what we were able to do was to, rather than the virus particle looking like Binjari virus, we were able to tell it to look like one of the pathogenic viruses instead. For instance, West Nile virus in this particular example here. So essentially what we were doing was making chimeric viruses. And I know that for some of you that may be quite alarming. Oh my gosh, what is she doing making these chimeras? If you look at the meaning of chimera in Greek mythology, you will find that it is a monstrous fire-breathing hybrid creature composed of different animal parts. And so yeah, essentially yes we are making hybrids, but what I want to emphasize here is that these hybrids or these chimeras that we're making are actually incredibly safe. So essentially what we were making is what we then coined the term a sheep in wolf's clothing. So we had a benign virus, Binjari virus, that is the sheep. It doesn't cause any disease. It cannot infect humans or animals. So we've got that at the core of our virus particle. But then we are dressing it up so that it looks like the wolf on the outside, so that scary pathogen on the outside of the virus particle. And so just to reiterate how we're doing this is that we're taking the Binjari virus genome here and we're exchanging these two genes here that form that outer case of the virus particle and making it look like Japanese encephalitis virus or West Nile virus or dengue virus, for instance. But the rest of the genome there is actually derived from Binjari virus. So everything else, so all of these genes here, these code for the replication machinery. These are the instruction, this is the instruction manual of the virus to tell the virus how to replicate itself over and over and over again in the insect cells. So essentially what we've got here is just, we're changing this outer coding here, but inside this virus particle, everything else belongs to Binjari virus. And so importantly, what this meant was we've got a chimeric particle that can replicate just nicely in mosquito cells in culture, but it cannot replicate in animals. So I thought you might be interested in how we actually engineer these chimeric particles. So I've got these photos here of Professor Alex Kromick and Dr. Yin Sito, who were instrumental in not only bringing this technology into the field, but also assisting in the optimization of the procedure as well. What we use is this methodology called circular polymerase extension reaction. And so what we have is this DNA, so these DNA fragments belonging to Binjari virus here, and then we've got other DNA fragments that code for these structural protein genes of the pathogen of interest, whether that be dengue or Zika, for instance. So what we then do is we carefully stitch these together. So these are overlapping ends. It's a bit like a jigsaw puzzle coming together, highly, highly specific. So we stitch these together in a tube with some various enzymes, and then we force this genetic information into mosquito cells in culture. After which we incubate these cultures for about 5 to 7 days, and sometimes with a lot of luck, we get these chimeric particles being secreted from these cells into the culture fluid in these dishes here. So now that we've got this system fully optimized, what we have found is that we can produce a brand new chimera in as little as 2 to 3 weeks, which is ideal for disease outbreaks. So just to highlight how robust this technology has been, we have produced so many different Binjari virus chimeras and dressing them up to look like a variety of different pathogens on the outside of the particle. So we've got chimeras for dengue, Zika, West Nile, yellow fever, Japanese encephalitis virus. So a lot of the viruses on that this phylogenetic tree over here, we likely have a chimera available for those viruses. So then we started looking at the features of our Binjari virus chimeras. And what we saw was that these chimeric particles had the incredible replication in mosquito cell culture, very much like Binjari virus. So if we look here, so basically what these graphs are showing is the number of virus particles that have been produced by these mosquito cells in culture. We're measuring how many of these virus particles are being secreted into this cell culture. So what we were seeing was that if we took, for instance, the pathogenic Zika virus here, this black line here, and compared it to our Binjari Zika chimera, we were getting 250 times more of our Binjari Zika chimera in comparison to pathogenic Zika virus. If we look at our Binjari dengue chimera, so dengue virus doesn't naturally replicate very well in cell culture. And so we were very pleased to see that our Binjari dengue chimera, we had 1,000 times more of our chimera being produced in culture. And so this is really important in terms of if we're looking at using these chimeric particles for vaccine or diagnostic applications, because what this tells us is that if we were looking at our Binjari dengue chimera, for instance, we only need 1 flask of our growing Binjari virus chimera. In comparison, if we were looking at growing up the wild type or the pathogenic dengue virus, we would need 1,000 flasks to generate the same amount of material if we were going to look at it being used in a vaccine capacity. Importantly, and as I mentioned before, we saw no replication of our Binjari virus chimeras in vertebrate cells in culture. So this is just a very small snapshot of a very extensive panel of animal cells that we have tested our chimeras in, and they do not replicate. And we've also shown this in animals as well. So we have vaccinated animals with our Binjari chimeras and have shown that in these animals that they do not replicate. So they're incredibly safe to handle. And I just wanted to highlight here that we've actually done a lot of work in trying to understand why our Binjari virus chimeras don't replicate in vertebrate cells. And there's actually, it's multifactorial. It's not just one reason why our chimeras don't replicate in vertebrate cells. One of the key things is temperature. The Binjari virus chimeras don't like replicating at 37 degrees. And another thing is that because Binjari virus evolved with mosquitoes, it hasn't had to evolve any of these mechanisms in which to evade the very sophisticated antiviral responses that naturally occur in vertebrate cells. And so when we've looked at these antiviral responses, the Binjari virus chimeras just can't evade them at all. And so therefore, they cannot replicate in those vertebrate cells. So one other very important feature of our Binjari virus chimeras is that we have shown that they are structurally indistinguishable to the pathogens by cryo-electron microscopy. So cryo-electron microscopy is where we use very high intensity microscopes to be able to visualize our virus particles. So here are our Binjari West Nile particles here as visualized under these super strong electron microscopes. So this was work led by Associate Professors Dan Waterson and Fasali Coulibaly, whereby they took matching cultures. So they grew up the pathogenic West Nile virus here, so the wolf, and they also grew up our sheep in wolf's clothing, so our Binjari West Nile chimera. So matching cultures of both of these. They purified them and then looked at them under the electron microscope and then resolved the structures of how these virus particles were looking on the outside. And so what they showed when they looked at these is that these particles actually look identical. You cannot tell the difference between, well, just on the outside of the particle anyway, whether it is the pathogenic West Nile virus or the Binjari West Nile virus chimera. And so why this is so important is that this system now enables us an incredibly safe way to resolve the virus structures of a number of different pathogens. So a lot of the flavivirus pathogens we don't have structures for because some of them are just too dangerous to handle, in large cultures that is. And so we've now got this incredibly safe system for researchers to be able to elucidate those virus structures. These structures also enable us to facilitate the development of better antivirals, better vaccines, therapeutics, and also better diagnostics as well. And importantly, this high level of structural authenticity makes these chimeras very good vaccine candidates, because it means that if an animal or a human, for instance, is vaccinated using these particles, that the body is seeing these particles exactly as it would the pathogen. So of course, we then went down that track and we have looked at a number of our Binjari virus chimeras in laboratory models to determine their effectiveness as a vaccine candidate. And ultimately what we showed is that for these chimeras that we have assessed, that we can provide a single dose of our Binjari virus chimeras and provide protection against pathogens in these laboratory models. So you can imagine our interest when about February last year, it became apparent that Australia was in the grips of an outbreak of Japanese encephalitis virus. So this was very much unprecedented, and I will tell you a little bit more about this outbreak in a moment. So of course, we wanted to then make a Binjari virus chimera that looked like the outbreak strain of Japanese encephalitis virus, and that is essentially what we did. Now, just to give a little bit of background about Japanese encephalitis virus, it does cause a very serious disease in humans and also in horses and pigs. And in Asia, it causes 68,000 cases of viral encephalitis a year. Thankfully, less than 1% of individuals that actually become infected with the virus actually develop disease symptoms. But unfortunately, if you do develop disease symptoms, there is, in 30% of these cases, it is fatal. And for the other 30 to 50% of individuals, there can be long-term neurological effects. So the natural transmission cycle of Japanese encephalitis virus is this waterbird mosquito transmission cycle here. So this is how it is naturally maintained in the environment. However, pigs can also become an amplifying host as well. And so pigs do develop enough virus in their bloodstream to infect a mosquito if it comes and bites them. And so there's another arm of this transmission cycle. Humans and horses, on the other hand, even though we become infected, we are classified as dead-end hosts because we don't develop enough virus in our bloodstream to subsequently infect a mosquito. So prior to 2021, the geographical distribution of Japanese encephalitis virus was limited to particularly Southeast Asia. And so we have had some minor incursions into Australia. So in the late 1990s and early 2000s, we had incursions of Japanese encephalitis into the Torres Strait, the islands of the Torres Strait between Papua New Guinea here and far north Queensland. And we also had a couple of incursions of Japanese encephalitis into far north Queensland as well. But the virus never took hold. We never had local transmission occurring subsequent to that. But then in early 2021, there was an outbreak, a fatal case on Tiwi Island, which is just north of Darwin. And this was of a particular genotype of Japanese encephalitis virus, which is a lot rarer. So this is genotype 4. And this was the first time that this genotype had been seen outside of Indonesia. So then fast forward to February 2022, and we get reports of an outbreak in piggeries in 3 states of Australia. And so this was very alarming. How did this virus get to 3 different states of Australia without being detected? And so this was also this genotype 4. So just to quickly mention why this outbreak may have happened. We've had, as you would know, we've had consecutive years of La Nina weather events. We've had a lot of flooding occurring over the last couple of years. And so what this means is that if we've got birds, water birds that are infected with Japanese encephalitis in these northern regions here, they actually follow these temporary wetlands that start to appear as these flooding events have taken place. And of course, huge mosquito populations that have also bred up as well. So what many people may not realise is that in Australia, we have got massive feral pig populations as well. And so this is a map here looking at the estimated feral pig densities in Australia. And so all of these together have likely fueled this outbreak. And so this is just to overlay here where we've seen the affected piggeries and also some of the human cases as well. And so all in all, we've had 45 human cases and unfortunately 7 deaths caused by Japanese encephalitis. And recent serosurveys in northern Victoria indicates that more than 3% of individuals living there have been exposed to Japanese encephalitis. More than 70 pig farms have been affected, and this has been quite devastating for our pig industry because it does cause very serious reproductive issues in the pigs. And horses can also become infected and develop symptoms as well. So we went ahead and we developed a Binjari virus chimera that looked identical to this outbreak strain of Japanese encephalitis virus. And what I want to point out here is that we currently don't have any veterinary vaccines currently available and approved for use in Australia. So we don't currently have any capacity to vaccinate our pigs or our horses. And this is because we've never needed it before, until just recently. So why do we want to vaccinate pigs in particular? This is because Japanese encephalitis virus in pigs causes substantial reproductive losses and also infertility in the pigs. It causes abortions and mummified fetuses in the pigs. And importantly, pigs also do play a role in the transmission cycle. So our aim, of course, was to develop a vaccine to keep the pigs healthy and to also stop this arm of the transmission cycle. So there's pig-mosquito-pig transmission cycle. By vaccinating the pigs, we want to stop that transmission cycle as well. Now, what I really want to emphasize at this point is that people cannot become infected with Japanese encephalitis from consuming pork meat and other pork products. The only way that humans can become infected with Japanese encephalitis virus is through the bite of an infected mosquito. So we went on to look at the efficacy of our vaccine in pigs. And so here is some data from unvaccinated pigs. And so what we see is when the pigs are infected with Japanese encephalitis virus, that usually over this 3 to 5 day period post-infection, that they develop levels of virus within their bloodstream that are detectable. And it is during these periods of time here, if a mosquito comes and bites these pigs, they will take up that virus in that blood meal and subsequently become infected. And it is then these infected mosquitoes, after a 5 to 15 day incubation period, if they bite a pig or a bird, a horse or a human, they can then transmit Japanese encephalitis virus onto those other animals. So in terms then of our vaccine trial, we went into weaner pigs, so young pigs, and these pigs were from sows that had not previously been exposed to Japanese encephalitis virus. And we vaccinated these pigs with one of 4 different formulations of our Binjari Japanese encephalitis virus vaccine. So these pigs received 2 doses 4 weeks apart, and it was of an inactivated vaccine. And so some of the pigs received a lower dose and some of the pigs received a higher dose. And then we monitored these pigs for protection against Japanese encephalitis virus infection. So when we looked at the antibody response, or how well these pigs had seroconverted, how good their immune response was against these vaccines, we showed that the majority of the pigs developed really, really good immune responses to our vaccine, apart from one pig here, and we'll see what happened with that pig on the next slide. So these were our protection results. So this is looking at how well our vaccine protected these pigs against Japanese encephalitis. So with our lower dose of our inactivated vaccine, we had complete protection. So none of these pigs became infected with Japanese encephalitis virus. However, only one of our pigs in the higher vaccine group did have breakthrough of infection, but all of the remaining pigs were negative. So this told us we had a high level of protection in pigs given also the higher dose of the vaccine as well. So all in all, this meant that we had 94% protection. So we're really excited about this, and the pig industry is very excited about this as well. If we can protect 94% of their pig population against Japanese encephalitis virus, and this is an excellent outcome. And so right now we've got another vaccine study that's occurring in older animals, just to prove that the older animals also develop good immune responses to our vaccines as well. And currently we're progressing towards commercial vaccine release, hopefully by the end of the year, so that we can really help boost the health of the pigs in our pig industry. So what I also wanted to highlight here, and I'm sorry that this slide is going to be a little bit busy, but not only can we use our Binjari virus chimeras for vaccine purposes, but we can also use them in diagnostic assays. And we actually partnered with Queensland Health for this work. So Queensland Health have a range of fantastic assays that they can test patients for whether they have been infected with flaviviruses such as dengue or Japanese encephalitis, for instance. And so what we've found is that on all these bead sets here, we can actually coat these beads here with our Binjari virus chimeras, and we can use them to determine if patients have antibodies against various pathogens. So if we look at some results here, so in one assay, we can simultaneously test an individual for a range of different diseases, whether it be dengue, Japanese encephalitis, or Zika, for example. And we've shown that we can actually use our Binjari chimeras effectively in these assays to accurately diagnose patients. And so Queensland Health have now commenced starting to roll out the application of our chimeras into their diagnostic systems. And it means that they can now produce these diagnostic reagents a lot more safely than using the normal pathogenic viruses. So now I just wanted to touch on some of the work that we've been doing in the context of West Nile virus. And so this is all about crocodiles. So West Nile virus is naturally maintained once again in a transmission cycle involving birds and mosquitoes. So humans and horses, for example, we can become infected with West Nile virus, but we don't participate in this transmission cycle here. Now, there are different strains of West Nile virus all around the world, and so the strains that aren't present in Australia are a lot more virulent, a lot more pathogenic than the strain of West Nile virus that we have in Australia. So our subtype of West Nile virus is called Kunjun, and so it is comparably benign, but it did actually cause an outbreak of equine encephalitis back in 2011. Now, in terms of crocodilians, West Nile virus in American alligators can cause death. And in Africa, in the Nile crocodiles, West Nile virus can cause substantial clinical signs. And so we were interested in, well, what does our local subtype of West Nile virus do in Australian saltwater crocodiles. And so it was actually really cool to be able to work with such a unique animal. So basically, the only thing that occurs in our saltwater crocodiles, if they become infected with West Nile virus, is that they develop what we term picks. And these are these little clearings here, these little lesions on the belly skin of the crocodiles. And so we wanted to look at whether we could then develop a vaccine for these. And the reason being is that crocodile farming is an emerging Australian primary industry, and so these lesions caused by West Nile virus can diminish the value of the skins during the tanning process. So with an amazing team highlighted down here, we looked at whether we could apply our Binjari chimeras to preventing Australian saltwater crocodiles from becoming infected with West Nile virus. So once again, we vaccinated these crocodiles 4 weeks apart. And what we showed is that regardless if these crocodiles received a low dose or a high dose, and regardless of, this is just the different methods that we inoculated the vaccine into the crocodiles, all of them developed a really nice immune response to our vaccine. Although we did notice that it didn't peak until around 7 months. So crocodiles being cold-blooded, their immune system's a lot more sluggish than a mammal. And so that's why we're not seeing the peak of these antibody responses until much later in the piece. So all in all, when we then looked at the effectiveness of our vaccine against preventing West Nile virus in crocodiles, we saw that the non-vaccinated crocodiles, 22% of these developed these PIX lesions in the skin here. Whereas our crocodiles vaccinated with our Binjari West Nile chimera, they had complete protection against West Nile virus infection. There were no lesions and no virus detected in the blood. So this keeps the crocodiles very happy. So we do think this is another success story here. And so just to highlight again that we do also use our Binjari West Nile virus chimeras in a diagnostic context as well. And so this work is being done by my PhD student, Ryan Johnston, who's in the audience tonight. So these lateral flow assays that are featured up here, I'm sure that most of you in the audience will have at some point in the last couple of years seen one of these guys here, the dreaded COVID-19 rat. So if you opened up one of these, you would see that inside there's actually a pad here that's got embedded in it gold particles. So these lines here that come up, it's actually gold particles that you're seeing on that line. And so they are coated with an antibody in this case here. So we've got an antibody here that's directed against crocodile antibodies. So what Ryan's been doing is then applying our Binjari West Nile virus chimeras to the test line here so that we can test crocodiles to see if they've become exposed to West Nile virus or not. And so he's generated some really beautiful data, so showing that if the crocodiles don't have antibodies against West Nile virus, that there's a negative result on these strips here. But as we get increasing amounts of antibody to West Nile virus, that we get increasing line intensity in our assay. And so, of course, the plan is to then translate this. We want to develop these for human assays, horse assays, pig assays, etc. And so now I just wanted to give you another very sneak peek at some of the latest data that's coming through. And this is being worked on by another PhD student in our lab, Michaela Bell. And she is working with another insect-specific virus, but this is an alpha virus. And so pathogenic alpha viruses include Ross River virus, Burma forest virus, and also chikungunya virus. And so basically we are replicating this system all over again. So alpha viruses do have a different genetic makeup, but essentially what we are doing is we are exchanging the structural protein genes of yada yada virus with those of the pathogenic alpha viruses, so that we are once again developing these chimeric particles that look like these pathogens on the outside, but have all of the very safe replication machinery of yada yada virus, the insect-specific alpha virus, on the inside. So watch this space. Michaela's doing some amazing work to really push this also out towards vaccine candidates and diagnostic agents as well. So this brings me to the end of my story tonight. So in summary, we believe that our Binjari platform does provide this recombinant vehicle for producing very safe and antigenically authentic vaccines. We can propagate these chimeras under minimum biocontainment. And so this then allows us to safely produce vaccines that may normally have to undergo quite high level biocontainment. We've shown that we can produce these chimeric particles from scratch in as little as 2 to 3 weeks. So this means that it is ideal for new disease outbreaks, and hopefully we don't have a reoccurrence of the last talk. I don't want any new flavivirus outbreaks happening within the next few months. We've got enough on our hands with Japanese encephalitis. We've demonstrated that we can protect pigs with our chimeric vaccines against Japanese encephalitis virus, and we've also shown that we can protect crocodiles against infection with West Nile virus. And so, of course, there's a huge number of people to acknowledge. I couldn't fit everyone on this particular slide here. And there's been enormous support from a range of different funding bodies over the years, both the Australian government research bodies, so the ARC, the NHMRC, but also a number of the partners with the Pig Project as well. So thank you very much to Australian Pork as well for some of the funding that they've provided us for the pig vaccine development as well. So I'm going to leave it there, and I welcome any questions about any of this work that I've presented.
Amazing, Jody, thank you so much. Question time. There's already some questions coming on the app, but if you haven't yet put one in, there is the QR code. Well played, IT team. You can scan that and get ready to ask your question. BrisScience will, of course, be back next month. Speaker to be announced very soon. And don't forget, this talk and all the ones previous, including the amazing talk from Tamara Davis last month, and our end-of-year spectacular science comedy show from last year is now up as well. A little bit more editing for that one. A lot of people and cameras. So make sure you hop online and see those. All right. Are you ready for some questions?
Yep. Okie
dokie. So first one, Ian asks, how does the virus impact mosquitoes? Is it asymptomatic?
We think so. So these viruses have naturally evolved with the mosquitoes, and so we don't believe that they cause any significant effect on the mosquitoes from which we've isolated them from.
Great. George asks, could humans spread mosquito viruses to other humans?
Sure. Well, there are some that you can. So I will give the example of Zika virus, for instance. So Zika virus can be transmitted from a mother to the baby in utero. So yes, it is possible to transmit from one human to another, but generally no, it's mosquito-borne.
Great. Seb asks, if the process for developing new vaccines in this way is so similar, would the approval process be accelerated or even skipped?
I can tell you that even getting these vaccines approved for veterinary use has been an incredibly hard road. So would approval processes be skipped? No.
Bad luck. Sorry, Seb.
Sorry.
Okay. Another question here. Sandy asks, how worried should I be about climate change and tropical diseases in Australia?
Yeah, I think that's a really big question. I think as there is more flooding events, as we've got more warming, it means that mosquito populations will naturally go to new areas. And so this brings mosquito-borne viruses to new areas of Australia. We're particularly worried about one particular mosquito species called Aedes albopictus, who has been knocking on Australia's door for many, many years, that we're trying to keep out of Australia. The problem with that particular mosquito species is that it thrives in cooler climates as well, and so it can transmit so many different mosquito-borne viruses, meaning that it may open up even southern regions of Australia to some of these mosquito-borne viruses that are typically only seen in the north.
So, I mean, aside from, I don't know, actually implementing climate policy in Australia, is there anything that we could or should be doing at this point? Like, how should we be preparing or thinking? Any advice?
No, that's really hard. I mean,
that's depressing.
Yeah.
All right, let's go to another question then. Freezing viruses. Seb, maybe same Seb. Is there a special way to freeze viruses so their body doesn't burst, or is that not an issue with viruses?
So we typically freeze them in the cell culture supernatant that we use. So they actually don't. Yeah, sometimes they can burst when you're freezing them, but it's not. They're not as susceptible as if you were freezing cells, for instance.
Right. James is now asking the question on everybody's lips: would this work with COVID-19?
No. So this for the Binjari platform, it is very specific for flaviviruses. We do have some other platforms in the pipeline, but this particular platform is not suitable at this point for something like COVID.
Okay. Couple more, and then we'll go get food.
Sure.
Right. Craig asks, if mosquitoes themselves become infected with one of these chimera viruses, would they effectively become an automatic vaccinator?
That would be great. But as you can imagine, there is a lot of regulation around that. So do we want mosquitoes being released that are going to vaccinate people? I don't think a lot of people would be very interested in that happening.
I don't know, if I could have released a bunch of mosquitoes around COVID and got everyone to get vaccinated, yeah, I'd be up for that.
I mean, it would be great, but what I can tell you is that we've actually already done the experiments to show that it's not possible for, say an animal got vaccinated, a mosquito came and bit exactly where the vaccine's gone into. The mosquitoes can't become infected with our chimeras in that manner. So no, not in the current situation, no.
All right, we'll go one more question now. I mean, I really wanted to know how you give a COVID test to a crocodile, because that seems like a... But I'm going to go with Kate instead, who's asking the big questions. Do the grad students have to feed the mosquitoes?
So we actually don't do a lot of the mosquitoes. So all of the mosquito work that we do, the mosquitoes are dead in our freezers. So we just do all of the grinding up. So no, my students don't have to feed the mosquitoes. With our collaborators, yes, but not my students.
Well, if that's not a resounding, endorsement of going to study in this field, I don't know what is. Could you please join me in thanking our amazing speaker this evening? Thank you so much, Jody.
Thank you. Oh, thank you.
And we would love for you to join us for some food and drink outside, and we'll see you next month. Have a great night.